Our today's topic is structure of a model membrane. The plasma membrane consists of lipids and proteins. The fundamental structure of the membrane is the phospholipid bilayer, which forms a stable barrier between two aqueous compartments. In the case of plasma membrane, these compartments are the inside and the outside of the cell. Proteins embedded within the phospholipid bilayer carry out the specific functions of the plasma membrane, including selective transport of molecules and cell-cell recognition. Let's start with the structure. The plasma membrane of mammalian red blood cells, that is erythrocytes, have been particularly useful as a model for studies of membrane structure. Mammalian red blood cells, we know, do not contain nuclei or internal membranes, so they represent a source from which pure plasma membrane can be easily isolated for biochemical analysis. Indeed, studies of the red blood cell plasma membrane provided the first evidence that biological membranes consist of lipid bilayer in 1925. Two Dutch scientists, E. Gorter and R. Grendel, extracted the membrane lipids from a known number of red blood cells corresponding to the known surface area of plasma membrane. Then they determined the surface area occupied by a monolayer of the extracted lipid spread out at an air-water interface. The surface area of the lipid monolayer turned out to be twice that occupied by the erythrocyte plasma membranes, leading to the conclusion that the membranes consisted of lipid bilayers rather than monolayers. The plasma membranes of animal cells contain four major phospholipids. The fundamental structure of plasma membrane in the case of plant cells and animal cells is similar. There are some minor differences in the composition that I will tell you later. So, the four major phospholipids present in the plasma membrane of animal cells are phosphatidylcholine. Just look at this diagram. Phosphatidylcholine, phosphatidylethynylamine, here, this blue in color. Then phosphatidylserine, this part, and sphingomyelin, here, this yellow in color, which together account for more than half of the lipid in most membranes. These phospholipids are asymmetrically distributed between the two halves of the membrane bilayer. The outer leaflet of the plasma membrane, this is outside of the cell and this is inside, that is cytosol. So this outer leaflet, this part of plasma membrane consists mainly of phosphatidylcholine and sphingomyelin, whereas phosphatidylethanolamine, this part, and second is phosphatidylserine, this part, are the predominant phospholipids of the inner leaflet. Our fifth phospholipid, that is phosphatidylinositol, this part, this green in color, is also localized to the inner half of the plasma membrane. Although phosphatidylinositol is a quantitatively minor membrane component, but it plays an important role in cell signaling. The head group of the both phosphatidylserine, just look at this part, phosphatidylserine and phosphatidylinositol, this part, these red small dot-like structures. Just look at this. Head group of these both phospholipids are negatively charged. So their predominance in the inner leaflet results in a net negative charges on the cytosolic phase of the plasma membrane. In addition to the phospholipids, the plasma membrane of animal cells contain glycolipids. Just look at this part. This is glycolipids and cholesterol. This part, this black in color. So the glycolipids are found exclusively in the outer leaflet of the plasma membrane. You can see here. This is the outer part, and these are the glycolipids. Then, with their carbohydrate portion, means glycolipids, which are found exclusively on the outer leaflet of the plasma membrane, with their carbohydrate portion exposed on the cell surface. So they are relatively minor membrane components, constituting only about 2% of the lipids of most plasma membranes. Now, cholesterol, on the other hand, is a major membrane constituent of animal cells, being present in about the same molar amount as the phospholipids. So two general features of phospholipid bilayers are critical to membrane function. First is the structure of phospholipids, which is responsible for the basic function of membrane as barrier between two means aqueous compartments, because the interior of the phospholipid bilayer is occupied by hydrophobic fatty acid chain. The membrane is impermeable to water-soluble molecules, including ions and most biological molecules. Second, bilayers of the naturally occurring phospholipids are viscous fluid, not solid. The fatty acids of most natural phospholipids have one or more double bonds and make them difficult to pack together. The long hydrocarbon chains of fatty acids therefore move freely in the interior of the membrane, so the membrane inside is soft and flexible. Now, listen carefully. Cholesterol plays a distinct role in the membrane structure. Cholesterol will not form a membrane by itself, but insert into bilayer of phospholipids. I can see here. This is cholesterol. And with its polar hydroxyl group, which is close to the phospholipid head group, depending on the temperature, this cholesterol has distinct effect on the membrane fluidity. At high temperature, cholesterol interferes with the movement of the phospholipid fatty acid chains, making the outer part of the membrane less fluid and reducing its permeability to small molecules. At low temperature, however, cholesterol has the opposite effect by interfering with the interaction between the fatty acid chains. Cholesterol prevents membranes from freezing and maintains membrane fluidity. So, cholesterol is an essential component of animal cell plasma membrane. Plant cells also lack cholesterol, but they contain related compounds, that is, sterols, that fulfill a similar function. Next is membrane proteins. Most plasma membrane consist of approximately 50% lipid and 50% protein by weight, with the carbohydrate portion of glycolipid and glycoproteins constituting 5 to 10% of the membrane mass. Since proteins are much larger than lipids, so this percentage corresponds to about one protein molecule per every 50 to 100 molecules of lipids. In 1972, Singer and Nicholson proposed the fluid mosaic model of membrane structure, which is now generally accepted as the basic paradigm for the organization of all biological membranes. In this model, membranes are viewed as two-dimensional fluid in which proteins are inserted into lipid bilayer. So just look at this diagram. These are lipid bilayer and these all are proteins. So Singer and Nicholson distinguished two classes of membrane associated proteins, which they are called peripheral and integral membrane proteins. Just look at this diagram. These are peripheral membrane proteins and these are integral membrane proteins, and these are the carbohydrates. So peripheral membrane proteins were operationally defined as proteins that dissociate from the membranes following treatments with polar reagent, such as solutions of extreme pH or high salt concentration that do not disrupt the phospholipid bilayer. Once dissociated from the membrane, peripheral protein are soluble in aqueous buffers. These proteins are not inserted into the hydrophobic interior of the lipid bilayer. Instead, they are indirectly associated with membrane through protein-protein interaction. So these interactions frequently involve ionic bond which are disrupted by extreme pH or high salt. In contrast to the peripheral membrane proteins, integral membrane proteins, these are integral membrane proteins. These proteins can be released only by the treatment that disrupt the phospholipid bilayer. You can see here. Portions of these integral membrane proteins are inserted into the lipid bilayer. So they can be dissociated only by reagent that disrupt hydrophobic interactions. The most commonly used reagent that for solubilization of the integral membrane proteins are detergents, which are small amphipathic molecules containing both hydrophobic and hydrophilic groups. The hydrophobic portion of detergent displace the membrane lipids and bind to the hydrophobic portion of the integral membrane proteins because the other end of the detergent molecule is hydrophilic and the detergent protein complex are soluble in aqueous solution. Now, I will tell you little about mobility and extracellular portion of plasma membrane proteins. Now, membrane proteins and phospholipids are unable to move back and forth between the inner and the outer leaflet of the membrane at an appreciable rate. However, because they are inserted into fluid lipid bilayer, both proteins and lipids are able to diffuse laterally through the membrane. All proteins are not able to diffuse freely through the membrane. In some cases, mobility of membrane proteins are restricted by their association with the cytoskeleton. In other cases, mobility of the membrane proteins may be restricted by their association with other membrane proteins, means with proteins on the surface of the adjacent cells or with the extracellular matrix. The extracellular portion of plasma membrane proteins are generally glycosylated. Likewise, the carbohydrate portion of glycolipids are exposed to the outer face of the plasma membrane. Consequently, the surface of the cell is covered by the carbohydrate coat known as the glycocalyx, formed by the oligosaccharides of glycolipids and transmembrane glycoproteins. Part of the role of this glycocalyx is to protect the cell surface. In addition, the oligosaccharides of the glycocalyx serve as marker for a variety of the cell-cell interaction. The structure of the plasma membrane is same in the case of both animal and plant cells. It is semi-permeable barrier made of the phospholipid bilayer with embedded proteins, then regulating the movement of substance into and out of the cells. So there are some minor differences. First is cholesterol. Animal cell contain cholesterol, while plant cell have sterol, which serve as the similar functions. Another is animal cell have glycolipids, which are carbohydrates and attached to the lipids in their plasma membrane. Thank you.